Jetzt sprechen die Blumen. Ich habe eine Aufgabe für dich vorbereitet. Schau mal, was hat dieser wunderschöne Rittersporn mit eurem wunderschönen Institut zu tun?
Diseases like cancer or COVID-19 can affect you, me, all of us. And everybody would like to stay healthy. But we don't understand all diseases. Therefore, we at the Max-Delbrück-Center for Molecular Medicine try to reveal the biological foundations of how we get sick and what can help us recover. I'm Casper Silvis, a PhD student in the group of Kathleen de la Rosa, and our team studies antibodies and the cells that produce them, called B cells. Our research is not always easy to understand. So let me take you into this using this flower. It is the delphinium flower, and it is very attractive for bees with its beautiful color. However, bees are not only attracted to color and smell, but also to the electrical field that surrounds the flower. When bees are flying through the air, the friction between the air and the bee causes the bee to become positively charged. On the other hand, the flower is slightly negatively charged, and the bee can sense the flower through the charge interaction. This is incredibly similar to how many proteins interact with each other, where they recognize positively and negatively charged amino acids, the building blocks of proteins, on each other's surface. One of the main reasons I came from the Netherlands to the MDC is that I can work with my favorite proteins, antibodies. Let me tell you about a new finding of our group based on an old concept in immunology. You know what? Let's do a bit of role-playing here. I am a coronavirus and my face is the spike protein, the protein on the surface of the virus that is responsible for attaching to host cells. Now, you are the immune system and you're the police and you're hunting me, the criminal virus. You're very good at facial recognition and you have all the tools called antibodies to recognize thousands and thousands of faces to know who's naughty or nice. Now, suppose I am masked. Suddenly you have a hard time recognizing me, and therefore you're less effective at eliminating me, the virus. Our team found that COVID-19 patients with severe disease had abnormally high amounts of the SARS-CoV-2 entry receptor ACE2 floating in the blood, which can bind to the spike protein. So in terms of the metaphor, the ACE2 is the mask binding to the receptor binding domain of the virus, thereby preventing the B cells and antibodies from recognizing the virus. This finding inspired us to investigate this mechanism for other viruses that may become a global health threat in the future or for which no effective vaccines are available yet. For example, another coronavirus, MERS. Could this phenomenon be a reason why there is no effective vaccine for MERS yet? And how can we get around this problem?
So this is what we are going to do.
We try to answer questions like these in a collaborative manner.
Yeah.
Like many small flowers that make up one functional delphinium, we are a lab full of enthusiastic people working together. We try to contribute our part to global knowledge to help solve global problems. The more we know, the better decisions we can make.